Hi, everyone. My name is Phil Liu. I'm a third-year PhD student in Stanford's biophysics program, currently studying protein engineering. I work with antibodies and kind of engineering them to bind to specific targets and make them bind tighter.
Thank you for joining us today, Phil. Can you talk a little bit about your experience as a Science Olympiad competitor? What were your favorite events and where did you compete?
Yeah, so I started competing since middle school, and I also then competed through high school. I think my favorite events were Fermi questions and cell biology. Afterwards, when I went to college, I actually had the opportunity to kind of help out on the other side. We actually had a Science Olympiad club, and we were responsible in running the LA regional tournament as well as the SoCal state tournament. And so helping out as first a tournament volunteer and then event supervisor, and then kind of the planning team for the tournament was kind of a really amazing experience in terms of experiencing Science Olympiad from the other side, like writing the tests and kind of planning the competition day of. Yeah, it basically was, I've been doing that ever since throughout college and now graduate school.
I love that. I think that's a big part of a lot of our alumni's experience is that they love Science Olympiad so much that they stick with it. They don't want to leave. So we're happy to have our alumni help out. Can you tell me a little bit about your undergraduate experience? Where did you go? What did you study?
Right. And so for undergrad, I went to Caltech and I studied biology. I also did research there in Pamela Bjorkman's lab, working on kind of therapies against HIV. And so it was my specific research was designing antibody-based constructs or antibody-like constructs targeting the HIV kind of protein spike on the HIV virus.
And then you went on to graduate school and are studying kind of similar ideas, right?
Right. Yeah. So I still work with antibodies now in grad school at Stanford. I work with antibodies kind of targeting cancer now and hoping to basically get the immune system to target cancer through the use of these antibodies.
Interesting. So that's a new way to kind of target cancer, right? And as I understand it, the way that we have treated cancer over the last few decades has really kind of changed over time. Can you talk a little bit about what that used to look like and where we're going in terms of cancer treatment?
Right. Yeah. And so for almost 80 years, the three main pillars of cancer treatment were surgery, radiotherapy, and chemotherapy. And so those techniques have been around for a while. Surgery's been around for centuries, however risky that was, way back then. And radiotherapy kind of came around the turn of the 20th century with Marie and Pierre Curie kind of exploring X-rays to treat tumors. And chemotherapies were really developed around World War II, when studies that were analyzing the effects of, DNA agents or cell toxins were then basically used as chemotherapies. And so in recent years, starting around the 1990s, there has been an increasing interest in focusing on exploring and aiding the body's own immune system to fight cancer. And so the excitement of, the field of immunotherapy has not only permeated throughout the cancer research field, but also the medical community and wider public. Like internet searches for explanations of immunotherapy has actually spiked since the mid-2010s. And it's actually been featured in kind of more popular general magazines such as Time.
Interesting. All right. So tell me about your specific research and what you are trying to do in your lab as you're exploring cancer treatments.
Yeah, so normally our bodies detect and kill tumor cells arising in tissues all the time in a process known as the cancer immunity cycle. So typically, cells express particular proteins on their surface to identify themselves as quote unquote self. And so with the mutations present in cancer, those proteins deviate from the normal self that the immune system recognizes and tolerates. As such, there are particular cells known as antigen-presenting cells that can collect these abnormal proteins and then proceed to prime and activate other immune cells known as T cells. And so these T cells then go to the location of the tumor and then proceed to kill these tumor cells, which then releases more of these abnormal proteins and then starts the cycle anew. And so that's normally how our immune system kind of recognizes, identifies, and controls and eliminates cancer from our body. And so cancer through these immune systems, or when tumors grow, essentially that the cancer has then escaped regulation from this immune cycle. And so part of the role that my lab is interested in doing is basically repairing the immunity cycle and getting our immune system to basically eliminate the cancer and restore the body back to where it was before. And so the antibody-based therapies that my lab is interested in, the proteins and kind of therapies that my lab are interested in are kind of on the left half of the cancer immunity cycle in this diagram, better targeting the immune cells to the tumor and also allowing the immune cells to eliminate the tumor once they reach the tumor.
And as I understand it, some of the things that you're learning are also could be applicable to other diseases. So you had started with HIV, with doing some similar practices. Some similar things are happening around diseases like COVID, right? There is some kind of application to many different diseases.
So antibodies and your immune system, they can target a whole lot of different diseases. And so like, you can actually have antibodies that target viruses, as you mentioned before, cancer, and also there are antibodies now used for treatments in like autoimmune disease. And so that's definitely like in that case, the antibodies are actually like telling the immune system to calm down and not be too overactive. Since in autoimmune disease, it's your own immune system getting too hyperactive and attacking your own cells. And so definitely like the field of modulating your immune system to target particular, like whether it be outside pathogens or like cells in your body that's run amok in terms of cancer, or like having like protein constructs or antibodies that tell your immune system to quiet down. Like these are all like, like really, really relevant. And like kind of a newish field that's become, I guess, really in the past like decade or so, really kind of burst onto the scene.
Well, thank you so much for doing that work. It's really important, I know, to a lot of people all over the world. If students are interested in this field, what types of Science Olympiad events should they be looking at? What resources, what organizations should they be looking at? What should they be doing?
Yeah, so definitely a lot of the biology Science Olympiad events are fairly relevant. Cell biology comes to mind. Also, I guess protein design comes to mind as well. And so even chemistry events, actually, since a lot of times at this sort of science, it's kind of at the interface of biology and chemistry. And so you get a lot of kind of molecules bouncing together. And so a lot of the chemistry applications actually like chemistry events also apply.
Last but not least, Phil, what advice do you have for our Science Olympiad participants who are competing in the tournament, or have already finished their season and getting ready for next year? What do you think they should be thinking about?
Yeah, and so I guess this is broadly applicable in all of science, but definitely don't give up. Science is all about, trying and maybe falling on your face a couple of times, but then, getting up, dusting off and saying like, there's always tomorrow and like, I can still try again. And definitely that's one thing throughout throughout, high school, college, and now graduate school that I've learned is, it doesn't matter how many times you fail. It just matters like what you learn from it, and being willing to try it again.
Okay, well, thank you so much,
Yeah.
and have a great day. Thanks for joining us today.
No problem. Thank you so much.